whatever, a particular enzyme. Right, so enzymes are proteins. They are biological catalysts. Here's just, remember, a lot of proteins are globular proteins. But from our last exercise, you've learned now that using the word globular, it kind of is misleading. You know, I think, an old piece of chewing gum is kind of globular. You think it's just kind of a random glob. These are not random globs. These are highly specific globs with a very particular active site. And this is the substrate that enzyme acts on. It's a particular molecule, whether it be D-glucose, for example. It could be D-glucose sitting in there, and that is what fits in that active site, and that's why L-glucose doesn't fit. It's that specific. What's going to go in there? So these enzymes are incredible. They are proteins. They're biological catalysts. They are globular in nature. They got this kind of this conformation, but if you look at it carefully, there's a particular little active site there. Exquisite specificity for their substrate, the molecule they act on. Exquisite specificity. Here's an example, catalase. Catalase. Now you're going to see that these enzymes typically end with an ASE ending. Catalase. What does catalase do? It converts hydrogen peroxide into water and oxygen. Where'd the hydrogen peroxide come from? A metabolic reaction that's going on inside the cell. It's producing hydrogen peroxide. It is an oxidizing agent. The cell says, get rid of that. Now. Right now. And catalase does it. 40 million molecules per second. All right? There's a catalyst for you. It can handle 40 million molecules of hydrogen peroxide per second. One molecule of catalase. All right? So it's not like your body has tons of these enzymes. It does have. It can make over 10,000 enzymes. Every cell can make thousands of different enzymes. But it's not like it has millions of these enzyme molecules. It has them, a few of them here and there, and they are turning over substrate molecules very quickly. They're being used over and over again. Remember in 1305, a catalyst is a substance that speeds up a chemical reaction without being consumed by the chemical reaction. So the catalase doesn't get reacted. It just gets used to move this hydrogen peroxide into, so that's a very tiny substrate. That's your substrate. This is the molecule that acts on. Converts it into water and oxygen, which the cell can use. The cell can use the water and the oxygen. All right, there's going to be 6 classes, and that's one of the questions on your sheet today is you're going to find an enzyme and you're going to try to figure out what class it fits in. What class does that enzyme fit in? Oxidoreductases, transferases, hydrolases, lyases, isomerases, and ligases. Now before you just kind of say, oh man, speaking a foreign language, think about some of these. You can start thinking about what these do. These are being put in a category according to what they do. So this kind of an enzyme catalyzes an oxidation reduction reaction. Like this right here. This is an oxidizing agent. It's being reduced to water and oxygen. So that is an oxidation reduction reaction. So catalase would be an oxidoreductase enzyme. Transferases, just take transfer. Well, let's look at each one of these. And so they're named after what kind of thing they do, what kind of reactions they get involved in. Hydrolases, they hydrolyze things. We've looked at hydrolysis reactions. We said that when a protein gets broken down into its amino acids, it's undergoing hydrolysis. So digesting protein, you need water. Water comes in and it breaks that amide bond and it starts forming back the amine and the carboxyl group, and you get your amino acids back. Lyases, isomerases. They isomerize the substrate. So they don't add or subtract anything to it, they just change its structure. And then ligases. So let's take a quick example of each one. We're on oxidoreductases. So here is the enzyme. And that's usually where we put an enzyme in a chemical reaction, over the arrows. And when the arrows are going both directions, and I hope this is right. I think that's right. Sometimes it takes one enzyme to move it in this direction. It takes a different enzyme to move it back. So I'm going to assume, right offhand, I'm not sure that's right, but let's assume it is. I'm just telling you, that's not always the case. There could be two enzymes involved here. One to get this to this, and one to get these two things back to this. All right, so that's lactate dehydrogenase. And why is that? Because the enzyme is trying to tell you exactly what it does. It takes lactate and it dehydrogenates it. Takes those two hydrogens off of it and forms a ketone, and NADH. All right, that's niacin. That's basic niacin. There it is, the NAD plus, and now it's NADH, reducing agent right there. So that's lactate dehydrogenase. These reactions, I'm giving you these as examples. You don't have to memorize all of these. Concentrate on the fact there are 6 categories. Transferases. Methylation is an incredibly important process that's going on in the body, and that's what this is doing. Here's norepinephrine, PNMT. Why do they call it that? Because that's its full name. Phenylethylamine N-methyltransferase. All right? PNMT. All right, so there's your enzyme. And it's transferring a methyl group onto this amine right here. So you got an NH2, got a primary amine, now you got a secondary amine. Epinephrine. EpiPen. Adrenaline. All right, so it's methylated the norepinephrine. I guess nor really no methyl group. Now it's methylated. So it's a transferase. It transfers a methyl group onto that molecule. All right, a couple that you're familiar with. We were just talking here about breaking down. Now that's saponification. This is hydrolysis. A hydrolase, you're going to react it with 3 molecules of water. You're going to get glycerol, just like you got over there. But instead of getting the sodium salt of this fatty acid, you get the fatty acid. So you get 3 fatty acids and glycerol. And that's a hydrolase, lipase, a lipid enzyme. It hydrolyzes lipids. Peptidase hydrolyzes peptide bonds. Breaks protein down into amino acids. So that's hydrolases. And the lyases work with double bonds. So you can see this is going to, this is hydration of, this is chapter 11, hydration of an alkene, but now it's occurring in a biological system. Fumarate is being converted into malate by hydration of a double bond. And the enzyme is fumarase. So lyases either add or delete, react a double bond. Isomerases, here's 3-phosphoglycerate. So this would be like glyceraldehyde that had got phosphated. It's got a phosphate group on it. And what is this doing? It's just moving the phosphate group from carbon 3 up to carbon 2. So it's isomerizing that molecule. Those two molecules, the starting material and the ending material, are just isomers of one another. Same chemical formula, different structure. All right, I'm just motoring through these pretty quickly. And then ligases have a lot to do with the bonds forming to make RNA or DNA, carbon-carbon bonds, carbon-sulfur bonds, carbon-oxygen, carbon-nitrogen bonds. So you got DNA ligase. You're going to form, you got a carbon OH, so that's going to become a carbon-oxygen bond right there to form that phosphate on the DNA backbone. Ligases. And let's see. Okay, what do catalysts do? They lower activation energies. It takes a big amount of energy to get this reaction started. The enzyme is lowering that. It's getting the two reactants close together and causing them to react. They lower that activation energy, which speeds up the reaction. There are things that affect. Did that switch on me? No. There are things that affect enzymes. Typically, you raise the temperature of a reaction, and the reaction rate increases. That's showing linear. But here is the biological enzyme. It has a peak temperature. Okay, and it's right around body temperature for human enzymes. And think about it. Remember when we studied proteins, we talked about one thing that can denature a protein? What's one thing that can denature a protein? Temperature. Okay? So you can't just say, oh, I'm going to just keep increasing temperature and these enzymes are going to work better. Because when you denature a protein, enzymes are proteins, you ruin their functionality. What do? You ruin their shape. That glob becomes even more random of a glob. And it doesn't function anymore. It loses that pocket, that active site. Can't work. So there are optimum temperatures. Okay? And here it is, working at an optimum temperature. There is an optimum pH at which the enzyme will work. And let's see, I think, yeah. And it's not the same for every enzyme. If you're working with an enzyme in the stomach, it's tuned to work at a very low pH. All right? pH 1.5. If it's in the bloodstream, so running through the liver, you got really a lot of blood flow going through the liver. The plasma is a pH of about 7.3 to 7.4. It's right there all the time. So the enzymes functioning in the liver are functioning at that. The enzymes that are adding calcium or phosphate to the bone or taking it off the bone are operating in an alkaline pH environment. The bone with a whole lot of calcium is alkaline. All right, so there are different optimal pHs for different enzymes, depending upon what part of the body they're functioning in, or what organism they're functioning in. All right, we could go in a deep dive into really, I mean, this is a massive area of research and study right now, is enzymes and how to tune them, how to shut them down, how to activate them for various things, for various diseases and conditions, etc., etc., etc. But what this is kind of showing is that you got an enzyme and you have a substrate. So if you think of catalase and hydrogen peroxide, they're there. There's two things there. What needs to happen to form the new product, water and oxygen? Well, a bunch of things. There's a bunch of steps that goes through. First of all, you have to form the enzyme substrate complex. The hydrogen peroxide has to come in to the active site. And it may then, at that point in time, you got this enzyme with a certain shape. When it binds the substrate, it actually may change its shape. Actually, the binding of that may cause interactions that it changes its shape, goes into a transition state. It breaks the water molecule up. It maybe brings in 2 hydrogen peroxides and makes 2 waters and 1 oxygen. So all of this is going on. It goes through this transition state. It opens back up again, releases the products, and you're at the end right here. You've got what you wanted to form, and you have this enzyme back in its original state, ready to receive more substrate and do it again. And like I said with catalase, does that 40 million times a second. Okay, so that's a rough idea, a general scenario of what's going on. Yeah, and so there's your enzyme with all of its amino acid sequence, all of its tertiary structure, etc., forming this pocket. And yeah, here's an enzyme, here's a substrate. One view of it is that, yeah, the substrate is perfectly matches the shape of the enzyme. Another view of it is, like I'm saying, the substrate comes into the enzyme and actually adjusts the conformation of the enzyme. These are just crude animations of it, much more complicated when you'd actually look at the molecules. But just trying to depict here, you can have this situation here, an induced fit model where the substrate changes the enzyme. So you get this activated complex. And then perhaps what's happening, this might be a disaccharide. It's going to break that bond and release 2 monosaccharides and readjust back to its original shape. Just a couple comments on specificity, and then we'll get to our activity. Various degrees of specificity. Enzymes, what do I mean by that? So we might have this enzyme urease. It takes urea and converts it into carbon dioxide and ammonia. Urea. Now we come along with a different substrate. Remember, this is the substrate. We come along with methyl urea. We put a methyl group on here. And urease says, I don't see that. That doesn't fit. That molecule doesn't fit here. I don't work on that. All right? So that's what I mean by specificity. It is specific for particular substrates. Now, there's different ways of referring to specificity. Absolute specificity. Now, that would have been a case right there. The urease absolutely will only work on urea. All right? It says, nothing else fits in this enzyme except urea. That's absolute specificity. Group specificity. You might have an enzyme that says, this enzyme can add a phosphate group to almost anything. So it's group specific. Linkage, like a lyase. It might be that this enzyme can work on any double bond. All right, a particular linkage can hydrate any double bond. And then stereochemical specificity. All the time we've been talking about D and L glucose. Why? There are enzymes that are stereochemically, optically specific. They're specific for certain optical isomers. So this is what we mean by specificity. And you could talk about an enzyme and say, how versatile is this? Will this do this to any other molecule like that? Does it work on any monosaccharide or just glucose? All right, and then when you get into genetic conditions, genetic diseases, a lot of times what you find is that person genetically cannot produce that particular enzyme, like the PKU, the digestion of phenylalanine. A person with PKU can't produce the enzyme to metabolize phenylalanine. All right, that's a specific condition they have. You can deal with that as long as you know it. As long as you know that's the situation. Doesn't mean you can't eat phenylalanine. It just means you can't eat so much of it that phenylalanine starts building up in your body. You do need phenylalanine. Your body will put it into protein. But if there's excess phenylalanine, it's saying this person can't burn that, can't metabolize that, doesn't know what to do with the excess phenylalanine. All right, so get into your groups. Put both your names. You're going to only turn in one sheet here for your group. I am going to come by. I'm going to come by and give you. I'm going to give you a metal, copper, selenium, some metal. What I want you to do is start drilling in and hunting for a human enzyme that has this at its active site. Just like hemoglobin has iron at its active site, most of these enzymes have at the active site a metal. All right, so let me come by and get you starting to look at a metal. I'm going to give everybody a different metal. You guys can have copper. Cobalt. You want to work together? Selenium, magnesium. I'll bring the sheet by in just a second. Manganese. And I'd like you to turn this in today. Let me just get everybody Oh, here, two. Oh, you got everybody have a medal? Oh, let me get their sheets, Brianna. Here's what you got to fill out. No, a cofactor is some other molecule that is required.
So it has to be like enzyme containing iron.
Yeah.
So many things in there. Yeah. Nickel-based enzymes.
Fill in these aspects of
These are regularly metal enzymes. The enzyme is produced by the human body. And find the information below regarding the enzyme. Indicate what type of enzyme it is. 6 types listed below. So, it's a type of metal.
That's just common. Put too much stuff on it.
Amine oxidase, containing.
No, it's starting to sound. Yeah. Nancy, you might just heard they say it. See, sometimes they'll say it's an equal one bacteria or this or that.
MAN.
Yeah.
Covered containing amine oxidase are found in bacteria, fungi, plants, and animals. Microbes, enzyme-enabled various I guess, well, isn't
Yeah.
the structure of proper amine oxidase?
Let's try to see what's the reaction.
So this is was the inside the metal. And the metal could be like.
Would this be the catalyzed oxidation reaction of a wide range of biogenic amines, including neurotransmitter cysteine and xenobiotic amines?
Yeah, what does it do to the amine? Oh, see what's it doing?
So that's probably
Converting it to an element. Yeah, so that's what I would put as
The reaction?
example, the reaction.
So just put that in.
Yeah, this is what it's doing. It's an amine enzyme.